All right, this first slide is simply to remind you of the existence of cranial nuclei, cranial nerve nuclei. And so we're accustomed to thinking about those cranial nerves emerging at various points from the brainstem, but they originate within these, for the most part, bilaterally arranged nuclei in the brainstem. These are the medulla, the pons here, and the midbrain at the top. We've already pointed out that a nucleus in this context refers to a cluster, an organization of neurons of similar function. In this case, they all form the origin of a specific cranial nerve. Now, this image, and I think not this one, but this one, you can be looking at concurrently, the little Netter's image there. You'll notice that for most of the nuclei, not all, but for most of the nuclei, there are both efferent, depending on whether the cranial nerve is motor, sensory, or whatever. But there are motor components and sensory components, and they're not located in the same location. So cranial nerve I, for example, may have a different location for its motor nucleus than for its sensory nucleus. It's primarily a sensory cranial nerve, so the motor nucleus is going to be smaller. So that's point number one. You'll notice that these nuclei aren't necessarily spherical in shape. Sometimes they're very, very columnar, running almost the entire length of the brainstem. We'll call that point number two. We'll have enumerated points. Number three, you can see that for most of these, the nuclei are bilateral. There are a couple of exceptions over here. You don't need to memorize these at this point. This is just put in for thoroughness. But in general, they are bilateral, and the significance of that, and one way in which the cranial nuclei, we're really talking about the motor nuclei here, differ from the spinal motor tracts that we've already talked about in the physiology, is that if this is a if we're talking about motor nuclei, most of these motor nuclei are bilateral. They provide bilateral innervation to the I guess you would say to the what's called the corticobulbar system. It would be the equivalent of the upper motor neuron. Let me try it this way. Remember, in the corticospinal tract, you have an upper motor neuron and a lower motor neuron. It's one neuron that decussates the upper motor and then goes down to the lower motor neuron. For most of the cranial motor functions, you have two bilateral upper motor neurons. They're called, it's part of what's called the corticobulbar tract. We'll look at a picture of it later. And so the muscles are bilaterally innervated. In other words, imagine two upper motor neurons, one from each side, and both of those to varying degrees are responsible for contraction of the activation of the lower neuron. As I say, we'll look at some examples of that. I just sort of want you to appreciate the importance of these nuclei. I think when we talk about cranial nerves, they tend to be overlooked. So here's a nice little table summarizing the cranial nerves. What you need to know, be sure you know for the anatomy test, is make sure you know the Roman numerals, know the name, know whether the cranial nerve is motor, sensory, or both. And then remember your cranial nerves that carry parasympathetic or autonomic fibers. Remember those parasympathetic fibers are piggybacking on these. We talked about that before. And you have those little mnemonics you can use that. Now, we're looking at the same thing here. This is the Netter's image, and it gives you a little finer appreciation of these nuclei. Netter's always confuses me because he doesn't use the standard. I guess he is the standard, but the coloring is always standard. So in this image, the motor is the red and the sensory is blue. So here are your motor nuclei over here, and here are your sensory. So for example, if you look at the trigeminal nucleus, this would be the trigeminal sensory nucleus. This is the trigeminal ganglion, right? So the ganglion is peripheral. And you can see they're connected by a portion of the cerebellar pedicle. This sensory pathways coming this way, passes into the nucleus from the ganglion via the cerebellar pedicle that we looked at earlier. Over here is the cranial nerve 5 motor nucleus, and you notice it's not extensive and it utilizes only one branch, the mandibular, the lower branch. Remember, the mandibular branch is responsible for innervating the muscles of mastication, a couple other small muscles. So I think that's the main thing. I just wanted to make sure. Oh, and the other thing, we mentioned this, it's not terribly important for a gross anatomy course at this level, but here's the trochlear. We'll talk about the function of the trochlear later. I think we'll look at a little animation or acronym today. But notice it leaves dorsally and then proceeds contralaterally, unlike all the others. Proceed ventrally. All right, so let's just quickly run through some of the points that I think are important in this context. Cranial nerve 1 is the olfactory cranial nerve. Generally, in these little tables up here, I've included just about everything you need to know, both in terms of the anatomy and the function. And then in these slides, more for your interest than for the purpose of the test, although in some cases this will be important. I've included what you can expect if that cranial nerve is impaired in some way, if it's been damaged, otherwise not functioning properly. So as you look at our illustration here, remember we're looking at these olfactory cells that reside, or at least extend their little processes through the apertures in the cribriform plate of the ethmoid bone. And so that's what we're looking at here. This is where things get awfully vague, awfully fast. But these are these are olfactory receptor cells. They're specialized, like most of the special senses. But they're often classified as bipolar neurons too. I'm not too concerned about the classification. But in any case, you can see that they really have a receptor component down here. Then they have more or less a fiber that extends through the cribriform plate up to little what are called glomeruli up here. You can see little tufts of glomeruli. And there are many types of olfactory receptor. Let's just call them type A, type B, type C, and so on. And all the type A's will go to one glomerulus. Even if it has to go over here, all the type B's will go to their own dedicated glomerulus. Yes? Is that simulating like a divergent type of cascade effect? So you're asking if the path, I really can't say what this particular illustration is trying to show in that context, but you should know that virtually all primary sensory pathways are highly convergent. That's almost always going to be the case, whether you interpret this as that. And then, so these are the olfactory cells or receptors, whatever you want to call them. And then you have a series of what are usually called mitral cells. Mitral cells then begin the process of carrying the olfactory signal back to various parts of the brain. And you know, as is often the case with the special senses, there are some interneurons here that seem to facilitate assimilation, processing, initial processing of information. You have this in your eyes, you have this in your ears as well. I don't know what you call these. We might just call these helper cells or processing cells that reside alongside the mitral cells. And you can see these helper cells are receiving internet signals. This happens in the retina. It happens in the ear too. We'll talk about that later, but I don't want to get into that now. But so all this information that is being carried along that olfactory nerve tract back to the brain. We have another illustration that will show the regions, the olfactory and piriform cortex, and so on. We talked about the rhinencephalon. Rhinencephalon, that's again means rostral or beak, as you would see in a dog or a mouse. Yeah.
Can you elaborate on the glomeruli?
Yes.
Is that the same as the mitral cells?
No. Well, what the glomeruli are really aggregations of interconnections, aggregations of synapses between the receptor cells and the mitral cells and whatever other cells may be interested in what's going on there. You can see a few of these ancillary cells. But what we were saying about the glomeruli, boy, there are a lot of theories about how we smell and how we taste now. In fact, there's a considerable degree of relationship between the two. There's a book about a couple of years ago written by a woman who suddenly lost her sense of smell, and it wasn't the sense of smell that bothered. It's called anosmia. She lost her ability to taste, and she became suicidal. So we'll come back to it. But anyway, getting back to the glomeruli, I went off my tangent here. The glomeruli are just specialized, again, sort of synaptic regions, clusters of synapse. And they seem to be highly specialized by the specific olfactory receptor, such as there's a type A glomerulus, a type B glomerulus. But all that means is that any given receptor is generally going to send its afferents to one type of glomerulus, whatever the anatomical significance of that is. We don't fully understand. As I say, there are a number of different theories about taste and odor.
So in this example, where does the olfactory nerve actually terminate? Because you have the terminus.
Yeah.
Where is it that it actually stops?
Yeah, good luck with that.
Okay.
So here's another illustration. So here we can see our receptor cells, and we can come back. So you can see, and we remember we said the olfactory sense is unusual in that it doesn't directly go to the brainstem and the thalamus. It goes right to the limbic brain. And that's what we talked about, this intimate association between odors and memories, odors and emotions, and so on. But anyway, getting to Bob's question, you can see that some of the afferents do go to relatively higher regions. You can see one going up there to the thalamus, but the direct linkage, the most direct linkage, is to what's called the olfactory tubercle, or the olfactory, what's left of the olfactory lobe in primates. And then you have a succession of little associated regions down here, such as the piriform cortex. Piriform just means pear-shaped, and it's usually considered to be part of the olfactory cortex. Look at that. Look, by the way, you can see several synapses here in recurrence, right? The next stop is the amygdaloid. What do you know about the amygdaloid? Emotion, rage, relation, and so on. So once again, that's that juxtaposition of odor and emotion. Then the entorhinal cortex. Clinically, this is of significance to you for a number of reasons. The entorhinal cortex is the cortex that is most intimately associated with the hippocampus memory, right? Episodic and especially place memory. But what we know that happens in the entorhinal cortex is that, for example, the fellow who won the Nobel Prize for discovering place cells, and the place cells are these specialized cells in your hippocampus, or in this case, the entorhinal cortex, that enable you to have a precise mental map of this room, this building, this Fort Myers, or whatever your home is. In any case, these are the cells that tend to be lost first in Alzheimer's disease, or at least among the first cells to be lost. And remember, we also talked about the fact that a common premonitory symptom of dementia is loss of smell. So it all sort of fits together.
I've read that like the reason that you lose your smell in Alzheimer's is because those cells need to constantly be remade from stem cell and progenitor cells. So wouldn't the MAP cells also just be like remade, and then once you lose that ability, that you're just not making anymore?
You may be right. I didn't get into that. You're exactly right. This region contains one of those focal centers of stem cell activity, and that certainly seems to be important for olfaction, but I don't think we know why. I think your speculation may be correct, but I don't know that that's been proven. And I don't know that a loss of that capability has been associated with AD. It may well be. I haven't seen that before. Yes.
You say that it's not directly linked to the thalamus. Is that because there's a synapse before it reaches the thalamus?
Well, you see, this is the thalamus here, and it's going up here. It's the linkage, and we sort of had this gets into a long and weary discussion, but there would be multiple synapses in this pathway, whatever this pathway is intended to be. It's not, I'm not sure what they're intending by this, because it just sort of disappears up here. But there would be multiple synapses, so this would not be a direct pathway. Whereas this, you have far fewer synaptic barriers to the limbic. We'll just call it the limbic brain, understanding that's a bit of a fiction. And then, of course, from the entorhinal cortex back to the hippocampus. So I think that's all I wanted to say about that. The optic nerve, we'll have much more to say about this later. I think all I would say at this point, as you look at the illustrations here, we can see the optic nerve going back to the optic chiasm. And in general, it will be the case that about half of the visual information from each eye, crosses over. And then you can see a part, I think they show the lateral geniculate nucleus of the thalamus. All right, that's going to be one of its initial stops. And then from there, that information is going to go back, proceed from the thalamus back to the visual processing centers in the occipital cortex. You've got a number of visual cortices. We'll work all of this out later in terms of which portion of the visual field crosses over and which doesn't. For now, just be aware that the optic chiasm is where that crossing occurs. It's presenting a problem if you're keeping up with all of this technological development in virtual reality. The way we process visual information is creating problems for some of these developers. 3, 4, and 6. 3, 4, and 6 are the cranial nerves that move the eye. 3 does most of the heavy lifting. It controls the 3 of the rectus muscles. It controls the muscle that elevates the lid, the superior palpebral muscle. And it also regulates one of the oblique muscles, the inferior oblique. So it handles most of the work. Very, very important in this regard. By the way, we call these muscles I've just talked about, the rectus muscles, the obliques, and for example, the palpebral muscle, those are called extrinsic muscles because they reside outside of the eye and they're moving the entire eye. The intrinsic muscles of the eye, which is what these terms refer to, are the muscles that help you focus your eye and also regulate the size of the pupil. Now, purely speaking, the oculomotor is simply regulating those rectus and palpebral and oblique muscles I mentioned. But piggybacking on that, we had that table of the cranial nerves that contain parasympathetic fibers. Piggybacking on that are parasympathetic fibers. And you can see some of them here. They're sort of in these little red dashed lines and dotted lines. So we won't get into this right now in detail, but I just want you to understand what we mean by extrinsic eye muscles, again, the muscles that are moving the entire eyeball up, down, whatever. But because cranial nerve 3 contains or includes parasympathetic fibers, we should at least mention the fact that intrinsic muscles are involved. We have a couple of words up here, meiosis, which describes technically the ability of the parasympathetic nervous system to reduce the size of the pupil. For example, your ability to change pupil size in response to changing light conditions, that's almost entirely parasympathetic. But if I say there's somebody with a very menacing face looking at us back there from the door, I don't know what they're doing, but I feel very frightened. That might cause your pupil to become large as you turn around. That would be a sympathetic response, which is not technically part of that. And by the way, I see that person back. Don't be alarmed. I'm sure he's perfectly harmless. But mydriasis refers to that sympathetic signal that enlarges the pupil. Generally, I would think of surprise. And as I mentioned, the pupillary light reflex is almost entirely parasympathetic, but those fibers are passing along, it is in part along the cranial nerve 3. By the way, as you look at these autonomic pathways indicated by the dashed lines and the dotted lines, we can deduce, it's not labeled here, because I didn't bother labeling it, but we can deduce whether the dashed line or the, I guess it's a dotted line, we can deduce whether one is sympathetic or parasympathetic. How would we do that? You see a synapse, and where is it? What is this thing? It's mentioned, calls it the ciliary ganglion. Is that a parasympathetic or a sympathetic ganglion? Parasympathetic. So which type of fiber would synapse in the parasympathetic ganglion? Sympathetic or parasympathetic? Parasympathetic. So here we have a synapse. All right. So this is the parasympathetic fiber, but you see how complicated nature makes it for us. What does the sympathetic fiber do? Go right through it, although it doesn't synapse there. We'll worry about that later. I just want you to begin some awareness of the complexity of these pathways.
From the superior cervical ganglia.
Well, the sympathetic, yeah, exactly right. Yeah, excellent. And we'll have a little review at the end of this section about the main points for your test. We've already talked about them, but I just want you to begin processing some of this information. All right. Remember 3, 4, and 6. 3, 4, and 6 then are the cranial nerves that regulate or move the eye, the bulb of the eye. We've said the 3 does most of the heavy lifting. 4 is called the trochlear because this little pulley up here, this is the muscle that innervates right here, the superior oblique. And that superior oblique bizarrely runs through that little thing right there, and that's called a trochlear or a trochlear pulley. So that's how it gets its name. But that's the only muscle that it innervates. I'm skipping over 5 because we're going to look at 3, 4, and 6 because these are the muscles, these are the cranial nerves that move the eyeball. So 6 is called the abducent or abducens because the muscle that is innervated by cranial nerve 6 is the lateral rectus. And so the lateral rectus is going to abduct the eye. So that one's simple too. In fact, we'll learn a little mnemonic later when we get to this.
Now we move on to look at the 3rd, 4th, and 6th cranial nerves, the oculomotor, trochlear, and abducent. They're motor nerves. Between them, they supply the 6 muscles that move the eye, and also the levator of the upper lid. As we've seen, the oculomotor nerve arises between the cerebral peduncles, the trochlear nerve arises from the back of the midbrain, and the abducent nerve arises below the pons. The only opening that these 3 nerves pass through is the superior orbital fissure, but there are openings in the dura.
So maybe make a note of that. Be sure you know that all 3 share that characteristic as well. They pass through the superior orbital fissure. Again, another way to keep all of this straight.
Quite a bit further back.
What he's saying now is that even though he's talking about these openings in the skull, remember what's overlying, what's covering the entire interior of the skull?
Dura.
Yeah, the dura, the meninges. And so now he's pointing out that there are cases where the openings in the meninges do not correspond. This is anatomy after all. Do not correspond to the openings in the skull. So that's what he's pointing out now. It's not terribly important. I just want you to be aware that it is not always the case that there's a nice, neat opening in the dura where the skull foramen happens to be.
The oculomotor nerve passes through the dura just alongside the posterior clinoid process, which is here. The trochlear nerve passes through the dura here. The abducent nerve down here. To follow them, we'll remove the dura over this area. We'll also remove this structure that we'll see later, the trigeminal ganglion. This cavity that we've opened into is the cavernous sinus. In the living body, it's filled with venous blood. Within the cavernous sinus lies the internal carotid artery. The third, fourth, and sixth nerves pass forward in the lateral wall of the cavernous sinus. Here's the oculomotor, here's the trochlear, here's the abducent. All three nerves pass forward into the orbit through the superior orbital fissure, which
So that's another anatomical relationship they all share. They pass through the lateral wall of the cavernous sinus. And you have several pictures of that. There's a couple of pictures showing that, but I think probably I gave you 4 or 5 pictures showing that in the previous set of slides. And of course, at this point, you should be familiar with the anatomical relationship of the cavernous sinus to the pituitary gland, the clinoid processes, the hypophyseal fossa.
Would it be the intercavernous sinus, or would it be the cavernous?
No, it's in actually the lateral wall of the cavernous sinus.
Okay.
The intercavernous is going to be medial. That intercavernous is right there around that pituitary stalk. Here we're out the ways, and then these things run right along that wall adjacent to and slightly anterior to the trigeminal ganglion. It's all packed in there together.
is here.
Oh. All right, so that brings us to the trigeminal. And we've indicated that the trigeminal goes by a variety of names. They're calling it here the semilunar ganglion. It's also called the Gasserian ganglion. There's another one. What is it? I cannot remember the other one. In any case, it's sometimes referred to as the DRG of the cranium because it acts like a large sensory, a DRG along the spinal cord. But having said that, you can see there are 3 major branches, and you know the relationship between the trigeminal nucleus and the ganglion. Cranial nerve 5 is unusual in this regard. But remember, the third branch, the mandibular branch, is also a motor branch. It also has efferents contained in it. So the cranial nerve 5 is not exclusively sensory. It is predominantly sensory with the mandibular, including efferents to move the muscles of mastication. Chewing muscles. And the largest cranial nerve, basically affords sensation from the entire face and much of the mucosa. And then it passes through that cerebellar pedicle that we talked about between the nucleus and the ganglion. So you can sort of see the 3 branches. We talked about how they leave the skull. Make sure you know that. We talked about, for example, the foramen ovale, foramen rotundum. And over in the top right, something we don't need to worry too much about now because it's something you're going to need to know in the next steps, not so much on this one. You can see the region of the skin that is innervated, that is supplied by each branch. So V3 is not only a motor branch, it also affords sensation, tactile sensation, somatic sensation from the mandibular region as well. And then this is simply showing you that the anterior trunk of V3 supplies the muscles of mastication, the temporalis, the maxilla, the 2 pterygoids. All kinds of clinical conditions resulting from problems with the trigeminal. You've probably heard of trigeminal neuralgia. I'm not sure the cause of that is well understood, but extremely painful in patients that are afflicted with it. Again, cause not known. This is a person who had trigeminal neuralgia trying to express their pain artistically. So we'll talk a lot about cranial nerve 5 and cranial nerve 7. In fact, they'll keep coming up over and over again in the remainder of our discussion of the head. Cranial nerve 7 is primarily a motor nerve, and its primary function is to innervate the muscles of facial expression. And for the next test, you'll need to be familiar with most of those muscles. And you can see a lot of them here. this very handsome individual. We'll also see, though, and I think we'll have time to look at a couple of illustrations of this, that the facial nerve includes some piggyback parasympathetic fibers. And it also has I don't think I would call it small, but it also has a relatively less important sensory function. It provides or brings the sensation of taste from the anterior tongue. Again, this is more important for the next test, but it's worth keeping in mind, just for purposes of clarity. We call the facial nerve, cranial nerve 7, a mixed nerve because although it's predominantly motor, it also carries those parasympathetic piggyback fibers and taste sensation from the anterior tongue. This was what I was trying to explain earlier. You can see that, for example, in this individual where they're trying to describe Bell's palsy, in this case where it's related to the facial nerve, that the lower motor neurons that supply the muscles of the face, unlike the spinal tract, are duly innervated from the right side and the left side. And this is usually called the corticobulbar tract. And so you have bilateral innervation. That's all we were talking about before. It complicates, in some cases, understanding of the neuroanatomical basis of disease. This is referring to Bell's palsy in association with the facial nerve. I think it's probably correct to argue that that's the case, that there's some type of dysfunction, paralysis, or loss of transmission through the facial nerve in cases of Bell's palsy. But they're many different. In most cases, we don't know what causes Bell's palsy. It could be viral, it could be idiopathic, whatever. And you can see some of the possible consequences that you may not anticipate. Again, loss of taste from the anterior tongue, impaired salivation. There's a muscle that's regulated by the facial nerve that regulates to some extent the dampening of the middle ear ossicles. So you get something called hyperacusis. You've ever been very tired, very fatigued, and you can sort of hear the rattling in your ear. Yeah. Thank you. I thought that was just me. This is kind of amusing. Is this the one? Yes. It says the color lines show the distribution of facial muscles paralyzed after a supranuclear lesion. What should that say? Yeah, supranuclear lesion. And if it's supranuclear, it'd be supranuclear. As I heard it pronounced.
We'll move on now to look at cranial nerves 7 through 12. We'll begin with
This
7.
one I'm going to zip over here a little bit.
This is the
So here's, let's see. Actually, let me just say the following. Because remember, cranial nerves 7 and 8 are sort of bound together at the interior surface of the skull, passing through the interior auditory meatus or the anterior internal acoustic meatus, whatever you want to call it. And then they sort of branch apart. And then 7 will emerge just under the ear here at the stylomastoid foramen. Stylomastoid foramen. Of course, 8 goes to the vestibular-cochlear system. But they're bundled together initially. And that's what he's talking about here, the internal auditory meatus or internal acoustic meatus.
Here's the internal auditory meatus in the dry skull. It's a short tunnel with 3 openings. The facial nerve passes forwards through this one to enter its own bony tunnel, the facial canal. In the facial canal, the facial nerve has a complex course in the temporal bone, passing round the wall of the tympanic cavity and coming out behind the styloid process here. To get a view of its course, we've removed the front wall of the external auditory meatus along this line. We've also unroofed a small part of the petrous temporal bone here. To represent the facial nerve, we'll add this green wire to the picture. Entering its canal, the facial nerve passes forwards briefly, then makes an abrupt U-turn called the genu, and passes backward. To follow it, we'll come round to the outside and look into the tympanic cavity. The facial nerve passes backwards high in the medial wall of the tympanic cavity, above the oval window. It then turns downwards to emerge here at the stylomastoid foramen, just behind the root of the styloid process.
Anyway, I want to look at
At the stylomastoid.
So this is the auditory, external auditory meatus. We won't worry about this now, but notice that the facial nerve just gets involved in everything. So that long transit, it just blindly weaves its way or wins its way through a variety of structures where it has no business. So it's actually running through the auditory system as well. We'll see that later. It doesn't do a lot there, but I just wanted you. This is a nice image of how it just sort of says how you doing and keeps on going.
Styloid foramen, just behind the root of the styloid process. On its way through the temporal bone, the facial nerve gives off 3 branches: the greater petrosal nerve, which is an autonomic branch, a branch in the middle ear to the stapedius muscle, and a special branch, the chorda tympani, represented by this wire. The chorda tympani passes upwards on the inside of the tympanic membrane and leaves the cranium here to join the lingual nerve. We've seen already that the chorda tympani transmits taste sensation from the anterior 2/3 of the tongue. Now, we'll follow the trunk of the facial nerve.
That chorda tympani is really going to complicate our lives on the next test, so I just figured it won't be a problem here. But it also, he talked about it bringing the taste sensation. It also sends out the little autonomic signals to regulate salivation down there too. So it's just a mess. Yeah.
Excuse me, is the diameter of that tract the cranial nerve passes through much larger than the cranial nerve itself?
The opening, you mean, through which the
That long tract is like, I mean. there's a
yeah, all of
lot
that's going to be filled with mucosa connective tissue. But the bony opening, yes, would be much larger than the nerve. But the nerve is not just, there's no open space in there. That's what you're saying.
Yeah, could that be a source of a lot of the palsies that arise from that nerve? It's a long,
Yeah.
winding tract that a little thing could be open through.
Oh, that would be a good topic for research. I don't recall encountering that, but it seems plausible to me. Yeah. I think the longer the distance, the greater likelihood you have of issues. All right, that's all I wanted to do. Cranial nerve 8 we'll talk about later, especially in the physiology. This is the vestibulocochlear nerve that, again, entirely sensory for both balance and hearing. Both fascinating. Actually, you have, I think you're getting one of your clinical courses, you get into the relationship of the visual and the vestibular system at length. So we don't get into it very deeply here. The glossopharyngeal cranial nerve 9 has, again, a variety of functions, just sort of all over the place. The motor function to parts of the tongue. In fact, it also has sensory innervation to the anterior surface of the tympanum, for example. The sensory sensations from a portion of the tongue. Remember, your tongue affords both somatic sensations, feelings of touch, temperature, and pain, and so on. But in addition to that, it has taste sensation, and those pathways are entirely different. And again, you don't need to worry about that for this test, but it will come. You will need to know them for the next test.
That controls the other 1/3 of the tongue.
Yes, as I say, we're going to talk about the relative allocation or division of labor between the anterior 2/3, the root of the tongue, both with respect to taste and somatic sensation. But we'll defer that until the next one. The vagus we'll talk about extensively, really for the remainder of the course. The vagus will be in our nightmares from here on out because It provides most of the parasympathetic innervation, about 75% of parasympathetic innervation to the thoracoabdominal cavity, to the viscera. And it's the only cranial nerve that actually descends into the abdominal cavity. As I say, we probably ought to chuck this one because I think modern contemporary anatomists no longer believe that the cranial nerve 11 is really a cranial nerve. We'll just call it the accessory nerve, but as we pointed out before, yeah, it originates in the spinal cord, goes up to the brain, then creeps along the floor of the cranial cavity, leaves, and then comes back down to primarily innervate the trapezius and the sternocleidomastoid muscles. So it's really an impostor. It's only pretending to be a cranial nerve. Presumably, there's more prestige in that business. It's like having iTunes over Spotify. So this part up here where it says chiefly motor to larynx and pharynx, now that function, I don't know what, I never know whether to tell you what's going to be on your exams or what is reflected in the current literature. But that I should get rid of that because that now is attributed to the vagus. Now that function is now those fibers that serve that function join up with cranial nerve 10 and they never leave it. So that's that portion of the portion of the pharyngeal regulation is now ascribed to the vagus nerve. And then you can see the function over here. We'll talk more about this later, but it's chiefly the upper trapezius and the SCM. And you will learn in your clinical courses, it doesn't happen all that often, but the consequences, excuse me, of loss of innervation to those muscles, the loss of innervation to the trapezius, which can be considerable, and to the sternocleidomastoid. Key thing to remember about the hypoglossal is pretty quick, pretty easy. It basically just innervates the tongue. Both the hypoglossal and cranial nerve 12 innervates. Again, we make this distinction between intrinsic and extrinsic muscles. Anybody want to stab at that? Not so much with your tongue, but what we mean, how does an extrinsic muscle of the tongue differ from an intrinsic muscle of the tongue? Right? Remember the analogy with the eye.
One moves the position of the tongue, the others change the shape of it.
Excellent. You said it better than I could. The extrinsic muscles of the tongue move the entire tongue in various ways. The intrinsic muscles of the tongue change its shape so that you're able to speak and mock the people around you. But both, fortunately, are largely innervated by the same cranial nerve. All right, so I have a quick summary here, just points, because I know that A&S is confusing with anatomy and so on. So here are just some general points that I want you to keep in mind for this test. And then, as I say, this stuff will rear its head for the remainder of the course. You know that there are sympathetic ganglia in the neck. How many are there? 3. There are 3 sympathetic ganglia. There are no parasympathetic ganglia in the neck. There are several parasympathetic ganglia in the head. We normally say 4. We could argue about that. But there are no sympathetic ganglia in the head. However, there are sympathetic pathways in the head. Remember the cranial nerves, and I don't even know that I agree with this entirely, that carry the parasympathetic fibers, 3, 7, 9, and 10. I could probably add another one in there. Let's not complicate things. You can see the cranial parasympathetic ganglia listed here, and I think you should certainly know ciliary, only so that you don't confuse it with the superior-most prevertebral sympathetic ganglia. What is the superior-most prevertebral sympathetic ganglia? Cilia. Yeah, so don't confuse the celiac with the ciliary. Ciliary resides just behind the eye there. The Pterygopalatine, otic, and submandibular. These 3 are primarily for salivation. I just throw this little pupillary light reflex in here because most textbooks describe this as being dual innervation from the sympathetic and parasympathetic. I think it's important for you as a PA to know that it's almost entirely parasympathetic. And so I have the pathway, both the somatic components and the parasympathetic components included there. We may have time to talk about this later. But remember, this is the pathway, and where that arrow is going, cranial nerve 2 and 3, this is the pathway that enables you to automatically regulate your pupils when you will be doing this too. You put the light in the patient's eye and watch what happens to both of their pupils. It's that parasympathetic reflex you're monitoring. This is the sympathetic pathway, and there's still much debate about the importance of the sympathetic pathway. Where it will be important for you, and I think we'll talk about this later, is in a condition called Horner's syndrome, where this sympathetic pathway has been interrupted, and remember that's going to affect the ability of the patient to raise the eyelid, and also going to affect sympathetic tone. What is tonus? Activation of the sphincter muscle, or the intrinsic muscle of the eye. So that's the only relevance there. And then I want to talk about this some more. So let's take a break and we'll come back and talk about these other papers.